In this video we will discuss about this, how this MMP9 level are overexpressed and how it is related to the C-terminal fragment of the N-Cadherin. So as from the heading it is clear that it's CTF2 mediates MMP9 overexpression, but how the authors come up to this point we will discuss through the results that are presented in this figure. So previously they checked the effect of this N-terminal fragment of N-Cadherin on the MMP9 overexpression, and here they are checking the C-terminal fragments of the N-Cadherins and its impact on the MMP9 overexpressions. So first what they checked it they checked the expression levels of this CTF1 and CTF2 when they used the secretase inhibitors. So when they use the secretase inhibitors, both the CTFs are formed. However, when they use only a PMA, then at that time the CTF1 expression levels are almost negligible, but CTF2 expression levels are clearly visible. And when they did it for this secretase inhibitors plus this PMA treated cells, in that case the CTF1 signals goes up because the secretase inhibitors prevent the further cleavage of this CTF1. And the author claims that because of the high levels of the CTF1 in both the secretase inhibitor conditions, they are not able to see enhanced signal for the CTF2. However, CTF2 is localized to the nucleus. So in order to check that, they did the subcellular detections of the CTF1 and CTF2 in both the conditions. In cytosols, they are able to see presence of both the they are able to see presence of this CTF1 but not of the CTF2. And in the case of nucleus also, they see the similar things, that is CTF1 signals can be visible but CTF2 signals are not visible. So however, they are claiming that CTF2 is nuclear localized or localized to the nucleus, and that helps in the MMP9 overexpressions. So now they change the strategy, and now they have the two different kind of fluorescence signals within the cells, and how they developed the system, they co-transfected this luciferase MMP9 and also this GFP positive CTF2 into the NPC cells, and then they check the signals for the luciferase when they use secretase inhibitors. So when they use the secretase inhibitors, as we know from the previous results of the experiments that are presented in this paper, that when they use secretase inhibitors, the MMP9 expression level goes down, and this is what they have seen for both the cell lines, that is NPC039 and NPC076. Afterwards, they checked the expression levels of the CTF2 using the Western blot at different conditions, and they are able to see that when they overexpressed this CTF2, they are able to see enhanced signals for the CTF2, and when they overexpressed with PMA, the MMA levels are also gone up, and this is true for the NPC039 and also for the NPC076. This indicates that the CTF2 overexpression somehow related to the increased expression level of MMP9. And afterwards they quantified this, they checked the intact N-Cadherin signals also that are shown on the bottom side of the panel C, where we can see that when the cells are treated with or co-transfected either with the control or with this F2 fragment, in that case they are able to see that there is a reductions in the full length N-Cadherin signals. However, when they used this CTF2, they do not see a decrease in the full length N-Cadherin. So this is what they have quantified from this panel C, and simultaneously they also checked the signals for this MMP9, and they can see that MMP9 signal goes up when the CTF2 is overexpressed and is treated with PMA, compared to the case where it is only treated with control transfected cells are only treated with PMA. So this could mean that high MMP9 levels will cause reductions in this full length N-Cadherin that can be seen on the left hand side bar plot of the panel C also. and when they are not treated with PMA, they do not see any impact on this full length N-Cadherin. So this MMP9 overexpressions is somehow causing the cleavage, and it creates a cascade effect, that is CTF2 get nuclearized and it will further overexpress MMP9. And next, in the final panel of this figure, they check the expression levels of this EGFP, which indicates the CTF2 expression levels, and they are able to see that when the cells are treated with PMA, the CTF levels are the EGFP signals goes up compared to the EGF signals goes up where CTF2 is overexpressed, compared to the case where only empty vector is transfected. And this is true for for both the cell lines. It can be seen here in the panel D. So this in all together indicates that it's the CTF2 that gets nuclearized when CTF1 cleavage is disrupted, and that leads to the overexpression of this MMP9. So this is basically a kind of cascade effect which is triggered by when the cells are treated with PMA in this case. So that's all for this video. If you need to understand more